And give me a text if anything looks horrible. Thanks, everyone. Welcome to the Water, Wetlands, and Watersheds seminar. Is today our first? No, we had one last week. So our second seminar of the week, but we're very lucky to have with us today Dr. Natalie Nelson. Natalie is Florida born and raised, but now she's an associate professor in biological and agricultural engineering at NC State University. She's also a fellow at the Center for Geospatial Analytics. And her lab is called the Coastal Watershed Analytics Lab, and they focus on looking at sources and fate and transport of pollution, both in coastal and inland waters. So Dr. Nelson has her bachelor's and her PhD here from UF in Ag and Bioengineering. We shared the same PhD advisor, and we'll get to see him for dinner tonight. It's great to have you back here with us and sharing your work. And it's been great to be a co-author and a collaborator. So thank you very much. And let's see if we actually have you. Yes, okay, you are good to go. Yeah.
All right, well, yeah, thanks so much, David. It's such a pleasure to be back. Like, it really transports me back to ground school to be in this room. So I had really enjoyed just the ritual of coming here every week for the seminar, being able to attend in person. I know I can still participate virtually, but there's just something about being in this room that I think is really special. And so I'm glad to see that there's so many students participating, because I really benefited from the seminar series, and I've actually been hoping someone would recreate something similar at NC State. So we're trying to do a little bit of a copycat here. Huh?
Yeah, it could be you.
Well, I am, and then I referenced it, so that's what's up. Yeah. And so, like David already mentioned, my research team at NC State is focusing on issues related to non-point source pollution, and we focus on both inland and coastal waters, and a lot of times looking at the connections between inland water management and downstream water quality and estuary systems. And so you'll get a flare of that in this talk, but then also if you're interested in more of the coastal work, that's what I'll be focusing on in the next seminar. And so here then again, I'm going to be talking about connections between engineered water management systems and connections to downstream water quality. So to start at a high level, we can consider the fact that across the US, there are over 91,000 dams. And these dams are constructed for many different reasons, but one of those reasons is flood prevention. So in places where you can have issues with chronic flooding, we often use these dams to ensure that water stays in the places we would like it to stay in. And so in cases when you have dams that are constructed for flood prevention purposes, they're often going to be managed by the Army Corps of Engineers, and they'll use an operations schedule to dictate their management. And so I know that many of you are going to be familiar with this already, but this is the operations schedule for Lake Okeechobee. We have water level on the y-axis, and then this is showing 2 years of the schedule. So it goes from December 2023 through December 2025. And this line here, this jagged line, is the observed water level. And so the Corps will monitor the observed water level and track to see which of these management bands it intersects. And based on the management band that it's intersecting, they then have their flow charts with decisions that they make about how much water to release and where. And so what you'll see here also is that there's this harsh line where the bands are changing. And so that was the product of there being a new operation schedule that was adopted in August of 2024. So there have been several operation schedules over time, and they've increasingly been cognizant of various environmental processes, basically acknowledging the ecological significance of this lake. But flood prevention is still the first and foremost objective of these operation schedules. And so when you have a system that is being managed for flood prevention, we care about flooding in places where there's typically people, there might be agriculture, like there are these human intensive activities that we care about protecting. It's not a flood unless there's someone who's going to be affected negatively by that overflowed water. And so when you have a dam that's installed for flood prevention purposes, that means that you are likely to be in a human intensive landscape, which means that there's more likelihood for high nutrient delivery. Dams are almost always old, so that means that typically you're also going to have issues with legacy nutrient stores. And then they reduce velocities through the system. So because of that, you have higher residence times. And all of these factors come together then to create conditions where you often have many issues with algal blooms. And so as you start releasing water, that can lead to there then being exported nutrients and blooms downstream. And this is something that people in Florida know well. And even though we know about this well, in many cases, actually linking up some of these decisions regarding management to the downstream consequence, like quantifying that relationship, can be quite challenging. So while there are these sort of like anecdotal understandings of how exactly we have these releases and they have impacts on water quality, actually then going through the data and parsing out those relationships is still a challenge. So we've been wondering how we can use many different data types to help us understand if regulated water releases could be scheduled to avoid nutrient and bloom export to downstream waters. All right, and so today I'm going to focus on basically these two sub-questions. So the first is, how do nutrient concentrations in blooms and downstream waters respond to water releases? And can blooms in downstream waters be attributed to release events using data? And so I'm going to be using Lake Okeechobee as an illustrative case study. I know you've all seen this figure a nauseating number of times, so I'm not going to walk through it in detail. But I also present these slides elsewhere, and once you leave Florida, like not everyone is familiar with what has happened in South Florida. So of course, though, people here know that now we have a lot of water that's going to the west and the east of Lake Okeechobee, and I'm going to be focusing primarily on the Caloosahatchee estuary in this talk, but I'll also talk about some of our work in terms of the connections with the St. Lucie. So of course, Lake Okeechobee is surrounded by intensive agriculture, and we know that it has these chronic issues with algal blooms. And in particular here, I want to highlight how you can have the blooms that will pool upstream of the water control structure. So in terms of thinking about when you have the release of water through a control structure, there's clearly very high opportunity to then export these problems downstream. And so the first study I'm going to highlight here was led by Lise Montefiore, who was a postdoc at the time of this study. And so this was funded through a grant that was led by Dr. Kaplan, the CLUE project. And so here, with Lise, we were trying to understand how do nutrient concentrations in downstream waters respond to water releases. So this was basically an exploration of the data such that we could understand and start to characterize these relationships. And so the way Lise went about this is that she compiled the existing nutrient chemistry data and flow data for this system. So here we're looking at Lake Okeechobee. We have flow data collected at these two water control structures. So the flow data are continuous and date back to 1966. So we have daily records going pretty far back in time. Flow that's coming out of S77, we can attribute to being directly from Lake Okeechobee. So that's a direct discharge from the lake. I'll refer to that also as a regulatory release, since it was a regulatory informed decision to release the water. And then we have flow at S79. So here, this water control structure is at the head of the Caloosahatchee River estuary, and it's connected by the C43 canal. So flow that's at S79, it's a combination of the water releases from Lake Okeechobee, but then also water inputs from the surrounding basin, the basin that surrounds the C43 canal. So we can look at also the difference in the flows between these two locations to start to estimate, well, how much flow is coming directly from the lake versus then the contributing basin around the C43 canal. Then we also have nutrient data at S79. So this would be trying to understand then what are the nutrients going into the estuary at the head of the estuary. And so these looked specifically at ammonium nitrate, orthophosphate, and total phosphorus. We couldn't look at the total nitrogen because there was just too much missing data. So the data were collected weekly to bi-weekly. It's cut off here. I believe it was starting in 2010. And then she downloaded the data like midway through 2022. So that's the period of record for this study. All right, so in the next few slides, I'm going to be showing you cases when I'll refer to the watershed as being the dominant flow source, or Lake Okeechobee. And so that's us looking at, in terms of the total flow going into S79, so being discharged into the estuary, how much of that can we partition into being releases from the lake versus the surrounding watershed? And we can make those calculations just by summarizing the flow data. So here what we're looking at is the variation in the contribution of flows to the estuary from the watershed, so from that C43 canal watershed and Lake Okeechobee. So the yellow colors are from the watershed, and the blue means that Lake Okeechobee releases dominated the total freshwater inputs. So each column is a different year, and then on the top here, each row is a different month, January to December, and then here we have 3 different seasons. These are hydrologic seasons, so defined to be of relevance to South Florida. So we have a dry season, an early wet season, and a wet season. And so what I mostly want you to look at here is how the colors are changing over time. So as we go from left to right, what you can see is that historically we had more watershed input dominating the total flow into the estuary. And then in more recent years, we see much more blue, especially in the dry season. So that means that Lake Okeechobee discharges are actually now starting to account for a larger overall proportion of the total flow into the estuary, especially in the drier times of year.
Is that 50% more?
Yeah, so in this case, well, here, this is continuous. So if you see,
I see.
but in terms of, we do have the summaries of overall, I don't remember the total in terms of flow, like what the actual change was, but this was a significant trend, as I recall. And so we can attribute some of these changes to then the fact that there are these different operation schedules that have been adopted over time. So at the time that we published this paper, the latest operating schedule was in the final stages of review, and then it's since been adopted. And so over time, like I mentioned before, they've gone from primarily just trying to maintain water levels within a certain range to then there was growing awareness of the ecological importance of this system, such that now there is consideration also of bloom risk, but also, for example, making sure to send enough water downstream to not have hypersaline conditions form in the estuary. So they have to account for a lot of different factors. But interestingly, as they've grown more environmentally minded, the footprint of Lake Okeechobee discharges on the Caloosahatchee estuary has actually grown. And I think that is a little bit counterintuitive. And we can look at specific years to try and get a sense for what is happening here. So if we look at 1990 and 2006, so we can see as in 1990, this is when they were still using an operation schedule that was purely driven by only flood prevention. They're just trying to maintain water levels within a certain range. And we can see that here you have some instances, for example, over here where you still have these major releases, but overall it tends to be more so short-term, very large releases. And then what they migrated to more recently was having then instead much more frequent but smaller releases. And so they would refer to these also as pulses that would happen, and that would be again to try and prevent hypersaline conditions downstream. But the net effect of this is that there's actually then much more flow that's going down because they are releasing much more frequently. And here then, and I should have mentioned here on this prior slide, so I'll use this consistently. The blue dots mean that Lake Okeechobee is the dominant source of flow, and then the yellow is corresponding to those watershed inputs. And so you can see the watershed is still the dominant source in the wet season because that's driven by the rain patterns. And so how does this then translate into the nutrient concentrations that we see going into the estuary? So here we have concentration on the y-axis and then discharge on the x-axis. So these are the CQ plots for those of you who are working in hydrology. And so each panel is corresponding to a different type of nutrient: ammonium, nitrate, total phosphorus, and orthophosphate. And here the slope, the beta is corresponding to the slope. So if you were to draw a regression line through these clouds of points, what is that rate of change? And so the yellow is then telling us also that the watershed was the dominant source of flow versus blue is for Lake Okeechobee releases. And so there are a few findings that we can go ahead and take away from these plots. So the first is that in terms of the concentration discharge relationship for the nitrogen species, we can see that there's a positive relationship with ammonium, which means that as you increase flow, you're also increasing the ammonium concentrations downstream. So you have this mobilization relationship. Whereas with nitrate, it's much more mixed. So I'll show on the next slide, you'll see that sometimes it's positive, sometimes it's negative, although overall it's net positive. Whereas with phosphorus, it's chemostatic. We basically see no variation as a function of discharge. We do see, however, that there are higher concentrations of phosphorus associated with watershed dominant inputs as opposed to Lake Okeechobee dominated inputs. And I say that because the tops of these clouds of points are more yellow. So that means that the concentrations are higher when the watershed is primarily the dominant source of flow. So we can start to then look more specifically on a month-by-month basis to see then how are these relationships changing over time, such that maybe we can identify certain time periods where there is a more optimal release time that you would then be able to hopefully minimize your impacts on downstream water quality. And so here, these are the slopes from those CQ relationships, but now broken out by month. So we have a regression line for each month. also each season, which is shown here. And so if you see hatching on the bar, that means that the slope was not significantly different from zero. So we just take that to be zero. And so what we can see here is that there could be a time period to avoid releases to prevent downstream nitrogen loading. That would basically only be though in January and February and June and July. So if you avoided releases in this time period, you would avoid the times when you have the largest increases in downstream nutrient concentrations. But if you look at other time periods, like here, for example, in the fall and early winter months, you could have a dilution effect with nitrate, but you'll still be increasing ammonia. And so I think the results highlight that it's a really challenging case to try and avoid downstream water quality impacts, but there are some considerations that can be made in terms of the timing of the releases. So from this study, our main takeaways were that these historical changes in operation approaches and that schedule has ultimately modified the hydrology, especially to the Caloosahatchee estuary. And that operations could potentially be modified to prioritize releases when ammonium and or nitrate are diluted instead of mobilized by these high flows and high releases. But the changes in regulatory releases are not expected to have an effect on phosphorus concentration. So it would require additional interventions, and they have many of those ideas already in the works in terms of developing additional basins and things like this. And so then also as part of this larger project funded by the Army Corps, we were interested in being able to predict and anticipate when you could have algal blooms occurring within the estuary. So now trying to build on these nutrient concentration relationships and also then with the hydrology of the system, like how can we start to develop some predictive models? And so there's also chlorophyll data that's collected in the Caloosahatchee estuary such that we can use that information to then develop algal bloom models. And so that was the focus of Maria Elena Menchu-Maldonado's work. So she was a postdoc at the time and had tried to investigate then if we could use these historical relationships that were characterized through this earlier work to develop forecast models of downstream bloom risk. And here I'm not going to go into much depth, but I mostly just wanted to highlight that this earlier work that helped us to understand how there are disparate relationships based on the dominant source of freshwater flow to the estuary was ultimately critical for understanding that we would need to have separate models to predict downstream algal bloom risk based on those freshwater flow conditions. So the way Maria Elena, she had tried to do this many different ways, and then this was ultimately the most successful approach. So she had partitioned the data into time periods when the watershed was the dominant source of flow, and then when we had entirely regulatory releases as the freshwater source to the estuary. And she created regression tree models to also try and mimic that same flow chart process that the Corps really likes to use, and that practitioners typically like. So we intentionally kept the regression trees to having just 3 categories that they could classify algal bloom risk into. We weren't trying to predict the actual concentrations, but instead trying to predict if the risk was high, moderate, or low. And so that's what you're seeing here in these plots is the observed values, but then also the predictions, which are the colored points. So the observations are in black. And then the predictions are always at just 3 levels because we're not trying to do that continuous simulation. So you can see then when the model is predicting, and then we have separate models based on the dominant freshwater source. So that early exploratory work helped us to then develop more so like a decision support tool. All right, and so we've also been wondering to what extent can blooms in downstream waters be attributed to release events? And so here this was work that was conducted by Natalie Reynolds, who was a master's student at the time funded by an NSF graduate research fellowship. And we had worked closely with researchers at the EPA to understand how we could start to use, in this case, satellite remote sensing to help us piece together some of these relationships. And so as I'm guessing many people in here already know, there were states of emergency that were issued in 2016 and 2018 because the blooms in the St. Lucie estuary were so severe in these years, and it was attributed to releases from Lake Okeechobee. And it was really undeniable, right? Like, so there were people who were reporting that water is being released, and then they're seeing blooms that were really terrible, like referred to as guacamole thick. So you can see here how intense these blooms were. And so here, when I say that the motivating research question is, can we attribute the blooms to the releases? like people's lived experience of this already confirms that we can attribute these blooms to the releases. But what do the data tell us? And so the data actually don't tell you that the blooms were that bad during this time period. And so if you were just someone like in the year 2026, looking back and trying to understand what was happening in 2016 and 18, you wouldn't really be able to parse this out from the data, at least not the grab sampling data. So here we're looking at the chlorophyll A concentrations on the y-axis. So this would be the proxy of the greenness of the water and the algal biomass in the water. And all of the different points are the chlorophyll concentrations that are collected from these different sites. So here, this is the St. Lucie, this is the canal that's connecting over to the St. Lucie estuary. And I'm mostly going to focus on that green dot because that's right there where the canal connects up with the lake at the water control structure. And so if we look at the green points, so here the black line is corresponding to discharge, and the dashed line here, I believe is at 40 micrograms per liter, which would be like a bloom threshold. But 40 micrograms per liter is not going to lead to this. This is going to be like in the hundreds. This is really, really high. So 40 micrograms per liter would be like the water is looking kind of green, nothing like this. And so what we see is here the red is corresponding to the periods when we had a state of emergency. There is this one really high concentration that's before the state of emergency, but largely in these time periods when we know that these blooms were ongoing, the concentrations are not that high. They're not like this type of bloom high. And so that's just the nature of grab sampling data. With discrete data, you can have these instances where you can miss blooms, where you might be sampling outside of a surface scum, such that your data are really not comprehensively representing what's happening. And so what we wanted to know is if satellite remote sensing could help us to fill the gaps in what we have in the grab sampling data. And so Natalie had looked through to see what types of satellite products would be most suitable. And then we landed on Sentinel-3, which has really coarse spatial resolution, but it has spectral resolution that allows for you to estimate cyanobacteria concentration. So not just algal blooms, but cyanobacteria specifically. And that's what's of relevance in Lake Okeechobee. So this is when we started working closely with the EPA because they already have a processed satellite product called Cyan that we could then use to start looking as basically like a retrospective of the system. And so Natalie had taken then the satellite remote sensing data from this time period, and she classified each of the pixels as having a bloom, bloom possible, or no bloom. And this was based on established relationships that had been published previously. And so she classified each of the pixels and then looked at what we referred to as this area of influence right upstream of the water control structure. And so she defined the radius of this area of influence as a function of hydraulic characteristics of the water control structure. So we believe that this would be approximately the area where you would expect the water to directly contribute to it, acknowledging that that varies as a function of flow. And so she looked then within this area of influence, how frequently did we have cases where the pixels were bloom or bloom possible? So like what is the total potential bloom area within that area of influence, and then looking at that over time. And so here's what she found. So there's a lot happening in this figure. I'm going to break it down. So first we have flow on the y-axis, and we're looking at different years, but as a function of water year instead of calendar year. So the states of emergency happened in water years 2017 and 2019. And so we have the discharge, but then here on the bottom, each of these different squares is corresponding to 1 month where we've summarized what was the total potential bloom frequency in that area of influence. So just upstream of the water control structure. And so when you have colors that are more yellow or green, that means that there were in fact dense blooms just upstream of the water control structure. And so we can start to then piece together when the blooms were upstream of the water control structure and when they had releases, and we can use that information to help us parse out how exactly those states of emergency occurred. So what we see is that in our 2 years with states of emergency, that the water releases were occurring when blooms were by the outlet. So that's intuitive, right? That makes sense. But now the data are confirming it. So instead of the grab sampling data largely masking that happened, now we can see through satellite that is in fact what happened. So here, for example, you see really high bloom frequency in terms of what we observed over the period of record and really high discharges. So it makes sense then that bloom export occurred. But we can also see cases when similar situations were happening in terms of the algal blooms, but we didn't have that same level of export. So also here, for example, we can see that there was a release when there were no blooms by the outlet. Like this was the largest discharge within this study period, but we can see also that in this time period, the blooms were relatively low. And so as a result of that, there wasn't the same level of severe bloom export, and some of those impacts were mitigated. And then in these last 2 years, this was also then there was like a lot of really careful planning to try and not have bloom export because it had been so severe in 2018. And so in these cases, we can see that there were also times when there were blooms that were just upstream of the water control structure, but now we see negative flows, like they're putting water back into the lake. And so they mitigated the impacts of having then downstream bloom export. And so from this work, so Natalie had also done like a lot of comparison in terms of the conclusions you might draw about bloom frequency. It wasn't only looking at this retrospective. So she had also made some assessments about the types of conclusions you would draw if you're looking at graph sampling data versus satellite remote sensing data. But what we found is that satellite imagery can be used to help fill gaps in historical monitoring to make sense of these prior bloom events. So there's still a lot of challenges with using satellite remote sensing data to do this type of assessment. But if you can look at pretty coarse timescales, like if you can do monthly summaries and still have use out of that based on the type of question that you're asking, then the satellite data can be really useful. And this type of information can also be used to inform near-term water release scheduling to mitigate bloom export. And in fact, the Corps is working now with people who provide them with satellite estimates so that they can use this type of information. And in the latest operations schedule, they're now starting to build those processes into their workflow. All right, so then now I'm going to go a little bit beyond Lake Okeechobee. And what I've been showing here is like the types of stories we can tell with monitoring data. So looking at how we can start to look over long time periods to understand how hydrology is changing, characterize relationships with nutrient chemistry, understand bloom export. But we're relying on the data that are available at these existing monitoring locations. And sometimes the processes by which those locations are selected are largely opaque, right? Like we don't really know the process. There often aren't reports that might provide justification, and a lot of times it's opportunistic. And so one of the PhD students I'm currently advising has been really interested in knowing how representative these nutrient concentration monitoring stations are of different types of conditions. And so there are 2 papers associated with this work that I'm going to gloss over very quickly here. So just to ensure that you understand what I mean when I say a monitoring station location, this isn't necessarily like a dedicated little station, but it's just a place that you're routinely revisiting. It's often going to be at water crossings that are easily accessible. And so they might be co-located with a gauge station, but basically you need to have a person who can easily go out and collect a sample of water. So there has to be a trail or something like a water crossing to facilitate that. And we have hundreds of thousands of these stations across the country. And so here is a map that shows sites with at least 40 measurements of phosphorus or nitrogen over 10 years. So we would estimate this as being approximately quarterly or seasonal sampling, although we didn't look to see how the samples were distributed over time. And so here you can see each of these points, though, is corresponding to a place where you have at least 40 measurements over a 10-year time period. And your eyes can immediately jump to some pretty clear spatial differences. So for example, Virginia, you can see is like perfectly drawn out in points, whereas West Virginia is the opposite. You can see it because there are so few stations relative to its neighbors. And so if we just do some simple summaries of this data, what we find is that surface waters in about 34% of watersheds in the continental US don't monitor for phosphorus or nitrogen. But some of these places are in arid regions where you wouldn't necessarily reasonably expect that they would monitor. And then 18% have more than 10 monitoring locations within a watershed, which here is defined as a HUC 8. So that's pretty good to have at least 10 monitoring stations, but it is ultimately 20% of watersheds. And so what we wanted to understand though is we can see that there are these state level differences, but when we think about dominant nutrient sources, which is typically going to be through agriculture, whether through livestock manure or fertilizer, do we see that nutrient monitoring stations are effectively paired up with the places where you expect to have the greatest nutrient delivery? And so we can use the National Nutrient Inventory, which is produced by a team out of the EPA, Robert Sabo's group. And we can look then to see they've compiled these nutrient inventory estimates to basically try and know like how much of nutrients are coming in versus going out. And they've reported it in a really easy to use format. So here what we're seeing is fertilizer and manure inputs. We decided to just focus on agriculture given its importance as the primary source of nutrient pollution. And just briefly here, when you're looking at phosphorus and nitrogen, you can see that there are certain places that light up as expected. But then we took this information and Chris combined it with monitoring station density. And so now this is a bivariate map where the colors are changing as a function of, here I'm just showing you phosphorus. They pretty much were the same for nitrogen. For area normalized phosphorus input, and then also looking at the monitoring station density. And so when you see a dark brown color, that means that there is high agricultural nutrient inputs, but no monitoring stations, so 0. Then as you get to lighter brown, that means that there's high nutrient inputs, but you do have at least some level of monitoring in terms of the number of stations within that watershed. And so we could use this then to start making estimates about the places that are and are not monitored effectively, although you could start to argue what is and isn't effective. And what we found is that about 135 to 152 watersheds have high inputs for at least one of these agricultural nutrient sources, but no monitoring stations. So there's some question about what is low versus high, but no is still no, right? It's 0. So there, I don't think there's as much to contest that would be inadequate to understand nutrient delivery out of that watershed. And I'll also mention though that there are some success stories highlighted here. So in the paper, we also highlight some places that do have really high nutrient inputs, but they're also doing a really good job of them capturing those inputs through their monitoring data. And Chris then also had done an investigation to try and understand, well, how does nutrient monitoring station density vary as a function of social vulnerability? So here then trying to understand, like, well, especially considering the fact that if you need a really nice water crossing and you need to have maybe a clear path to a waterway, those seem like the types of characteristics that are going to be largely maintained by local municipalities. And so you would expect that the level of vulnerability within the community could affect the extent to which an area might be considered suitable for monitoring, which could in turn potentially lead to there being less monitoring in more vulnerable places. But that was the hypothesis and Chris investigated. So Chris pulled together all of the social vulnerability index data. So this is an index that accounts for many different dimensions of vulnerability. It's produced by the CDC and it was developed with the intention of informing disaster recovery. And so Chris had compiled all of the social vulnerability index with nutrient monitoring density. And so here he was looking at a shorter time period than what I showed for the national study. So the stations are slightly different, but still you can see that Florida, for example, like really high monitoring density, and then it's more sparse depending on where you are in this region.
15 minutes.
Perfect, sorry, I'm in good shape. All right, and so then what Chris evaluated is to what extent do the observed monitoring station densities match up with what would be expected if you had a perfectly proportional distribution of stations as a function of different characteristics. So for example, if we account for area, so just hypothetically, if you had like 10% of an overall region in the most vulnerable category, you would expect then that 10% of monitoring stations would be in those areas if you had this perfectly proportional relationship. And so if you look then at the difference between the expected distribution under perfectly proportional conditions versus the observed, that's the deviation that's shown here. So you have deviation on the y-axis, and then this is the social vulnerability index that's divided into deciles. So it goes from 0 to 1. And so 0.9 to 1 would be the most vulnerable places, and then the low vulnerability would be 0 to 0.1. So you can think of this also as being approximately a proxy for like more affluent areas and low vulnerability versus poor areas and high vulnerability. And what you find is that if you account for different factors like area and population, but also the amount of water area. So this was estimated as the flow line length. So this would be like the total linear distance of streams, but also the perimeter of polygons like lakes. If you take that total distance and if you correct for these different factors, what do you see in terms of the distribution of where monitoring stations are located? And so typically then the more affluent areas have more monitoring stations than what you would expect if you had then this perfectly proportionate relationship. But then the higher vulnerability areas tend to have fewer stations than expected. And so this is across the region. But if we also look more specifically at Florida, you see that that same relationship holds. So I think this is valuable to consider because these are showing different forms of bias in terms of where nutrient monitoring stations are located. And so you have to account for this when you're running different types of data intensive analyses. So especially in this time when we have a lot of machine learning models that are being applied at really large scales, we often have then these constraints of where data are collected, and that's fine, but then acknowledging that there could be these sources of bias and that some places might not be well represented. And so in terms of thinking about vulnerability, that's related to the impact of your work. And then in terms of the agricultural nutrient inputs, that's also related to like, are you even capturing some of the most dominant sources within your data? So when telling stories from monitoring data, particularly at these large spatial scales, you'll want to consider which communities aren't represented. All right, so back to this original question then that I opened with, which was, can regulated water releases be scheduled to avoid nutrient and bloom export to downstream waters? Yes, but to a limited extent, especially for phosphorus. So with nitrogen, it does seem like in the case of Lake Okeechobee specifically, there could be some opportunities, albeit still limited. And using diverse monitoring data helps to parse out optimal time periods for issuing releases, but it also helps us to start connecting different pieces of a story that we're often hearing about, but then don't have really well represented with some of like individual data sets that are out there. So bringing these together can help. And when analyzing monitoring data, it's important to be mindful of potential bias in sampling locations and at least just considering how that could potentially influence your interpretation of your results. So that's it for this presentation. I'm happy to take any questions.
Great, thanks. Megan, if you'll help with the YouTube questions, we'll start in the room, and we always like to start with students because we know the professors can come up with questions for days, but so any questions for for Natalie? I'm gonna get a picture of this full room.
Sure.
You can just put that in your annual report.
Shots of me.
Yeah, exactly. You can put it in your like annual report. There you go.
Yeah.
Okay. Questions for Dr. Nelson? Yeah, please.
I was just gonna ask, what did you do your doctorate in when you were here? Was it water related or?
Yeah,
Okay.
yeah. So I was in the ag and bioengineering department. My advisor was Rafa Munoz Carpena. So like David said, we're leaves of the same academic tree.
Mhm.
And I had focused on harmful algal blooms. So trying to understand bloom dynamics in a few different systems in Florida. So Lake George, Florida Bay. And I had also looked at dissolved oxygen in tidal creeks as a sort of aside that became part of my dissertation.
Yeah.
So, if you can't use the like release timing to control harmful algal blooms, is there any attempt to like reduce sources or otherwise use the nutrients? Like evidently they're really good at generating algae, and there's a big focus on like biofuels from algae. Is there any way to like partner maybe with like the industry that's trying to grow algae to use like Okeechobee water and then return the water with less nutrients on the other side of the system to then decrease the source concentration, but would then decrease?
Yeah. Like
Repeat
maybe
the
that's
question
just-
real quick. So if you can't control release timing to reduce nutrient loads downstream, what can you do to reduce it in other creative ways? So.
Yeah, it's a great question. And so I haven't done any research in that area, and so I don't really feel well equipped to provide a good answer. But I can tell you that I know that there's private industry that is doing something related to this in Lake Okeechobee. I just don't know the details. So certainly there is interest in these types of like, I guess we call it kind of harvesting algae as a nutrient removal strategy. I mean, they remove shad in lakes as a nutrient removal strategy. So there's a lot of interest in those types of approaches. But I just don't know enough to tell you if I think that seems feasible or could be done at scale in a meaningful way in Lake Okeechobee.
Go ahead, Dr. Yule, yeah.
Just a comment there, I think it's not considered a good idea to develop an industry around something you're trying to get rid of.
Like if you could, ideally, if you could make agriculture not use as much of it so that it doesn't bleach, like that would be fine. But if you're already assuming that agriculture is gonna put it there, then you wouldn't be able to use it in some other way rather than just having to flow through the system.
Yeah, I will note that the Army Corps is funding work that has in situ algal collection, harvesting, and then, cleaner water going out the other side and using it for biofuel production. It's at like a very pilot scale, but so successful, but the scale for Lake Okeechobee, that's always the question is what's the scale? We have a question. We have a question from YouTube, which is the 2020 water year release pattern was quite different. Given that it coincided with the pandemic, I'm curious whether related changes in operations or human activity in 2020 were considered.
We did not, so we really didn't try to, that's a great question. We didn't try to explain their release patterns. I guess that I had surmised that there could be connections with the whiplash of like having gone through this really public event with having extreme blooms, such that in the following years, there may have been more interest in then making sure, like especially that there weren't exports if they knew that there were blooms. But I would need to pull more data to understand. Do you know the answer?
No.
Oh, okay, because I would also need to know, for example, I don't remember now, like, was that a dry or a wet year? Because in some cases, when they're back pumping, there could be other reasons for that aside from algal blooms that are more so driven by local waters.
Yeah, there were some very political, vocal political voices on the East Coast, on the St. Lucie side that were strongly, they were effective in reducing flows to the east, and they continued, well, they didn't quite get them in 2021, but it's been, yeah, I think that's been I don't know if the pandemic had anything to do with it, but politics definitely did.
I see,
And strong
okay.
representation from one of the Congress people.
Okay, yeah, and there had been a question in the
Yeah.
back there.
Yeah, go ahead.
So with the overlapping satellite data, I know that the Landsat 8 was what you used, correct?
We use Sentinel-3.
Sentinel-3, so that was a 300 meter square
Yes.
area.
Yeah.
So it's compiled to be almost 10 meters squared from the next one. So how did you, oh, those are kilometers. Okay.
Yeah.
Yeah, I don't think she rescaled I don't think she rescaled. Yeah. Yeah, but it's really porous, and so that also makes it extremely difficult to validate with grab sampling data, because how do you compare one scoop of water with the 300 meter by 300? So that's a lot of what this paper investigated, was looking at different measures for doing that comparison, like with frequency-based assessments, as opposed to direct matchups of concentrations.
AJ?
There are other students, sorry.
We can pop in.
Okay, cool. So it's kind of building off of this project, and it's really cool, really creative use of the remote sensing and coupling of the management decisions. But this kind of is assuming that the bloom that we see in the estuary is the same bloom that we see in Lake O, right? So do you, or is contributing at least? I'm curious if you have any information or if anyone's working on the processing and like remineralization, decomposition, denitrification, and fixate, all the stuff that we talked about earlier in that canal along that pathway, and if there are ways to maybe optimize those kind of benefits.
There's a whole project on that, but I'm not part of that one, so David, do you want to?
Well,
talk about that?
Well, I guess two things, and will just maybe real brief. One is that we did track toxic microcystis algal bloom down the Caloosahatchee side and sort of used modeling and graph sampling, and Ed Flips was involved and your team was involved, Luis, and so sort of watch what happened to the bloom as it traveled down. Did the toxins lyse out and all that kind of thing. On the east side, I'd probably throw it to Elise to, yeah.
Yeah, well, Mauricio, I guess, is leading the St. Lucie project that's a complement to the Caloosahatchee, and so part of Madeline's work is looking at that. And yeah, we could.
Down
talk
on
about
that,
it.
it's great. So, if there's anything else.
So you're gonna give the talk next week? In 2 years? In 2 years? Yeah.
You know, I think part of the challenge though is just like also when you look at the grab sampling data, yeah, it becomes hard to look at the routine monitoring data to start to even, I think, address
Yeah.
this really important question that you've asked. But yes,
Think
like,
about like the SUNA conversation and the DO conversation that we had, and we could think about. I have thoughts.
yeah,
Mhm.
all right, well.
But I think what's seen is it's like transport, like is there a bloom and it transports, that's one thing, but it transports and it changes, but it could also, you could transport like a little seed of
Mhm.
a toxic algal bloom into a nutrient soup.
Yep.
And it will grow, so is it, you know, this like.
Is it the nutrients,
Is it
is it the
allotment,
feed, is
is
it-
it all-
yeah.
it's like we get real nerdy real quick, but it makes a difference because does it matter whether you release it, and it doesn't. You could release a very algal-free plug of water that's nutrient rich with just a little bit of seed and you're suddenly, you're
Yeah.
seeding.
Yeah,
mixture.
exactly.
So I think we have time for like 1 or 2 more questions before we have another seminar down at 102. Yes, Rob?
Yeah, since the, no, at least for the St. Lucie side, since the problem arises by a canal, the C43 canal, why is that canal there in the first place? So what is the
For
current
the
benefit?
Lucie?
On
The
both,
C40
Yeah, the one that goes to the,
The east or the west, so the C,
oh, yeah, okay, towards the west.
This
Yeah,
one?
yeah, so there's a canal that transports like all the stuff to the estuary.
yeah.
But since it's a canal, so it's a man-made feature, but what is the current benefit of having that canal? Why do they need to like release water in that direction in the first place?
let's see if any, let's see.
History class about
Yeah.
I wanted to create the crossboard. What was it called now? Like there's a lot of history with that canal now. I think there is a lot of industry that's dependent on it.
So students, why don't they just send all the water south to the Everglades instead of mucking up to the east and the west?
Wasn't that to connect up the fish eating creek with Okeechobee? To take advantage of. the fish
But
eating
even
creek?
so, now the canal, why don't you just not put water in the canal? Just send it to the Everglades. Everglades is too dry, right? Okay.
I'll have to bring that.
Water quality, okay, so water quality remains an issue, so you can't, you know, you're under very strict regulation about the quality of water that can be discharged to the Everglades, right? Despite all the treatment wetlands that are built down there and optimized, so we're still not getting there, but, all right, one last question, AJ.
Okay, this might be a really dumb question, but I'm thinking back to what AJ was saying, and I think about Nick's research about like the red tide, like we know that lab doesn't necessarily translate into real life, but has anybody not looked at like if you have a certain, like if the nutrients are already there, then you add a seed, does it grow? If you don't, you have it backwards, does it grow? Has anybody ever looked at like the process of like what makes these blooms happen like in a lab?
Yes,
Okay.
yeah, and like, so, you know, Ed Flips is someone who's done a combination of lab and fieldwork, and there is good science out there. I think it becomes quite challenging to scale it to like an entire water body essentially. Especially when it's dynamic as this, where you have to account for human decision making as well.
Right, but then people like use that to kind of inform models and stuff like that.
Yeah, absolutely. So there are process-based models that account for many different processes that are influencing both the growth and loss of algae. And every level of complexity you can imagine, from the simplest to like a dizzying number of equations. Yeah.
All right, let's thank Dr. Nelson one more time. Okay, so our speaker next week is Dr. Corinne Matias. She's from the geography department here. She's going to be talking about hurricanes in Florida and probably around the globe. So thank you very much. I saw everyone's assignments are turned in. I'll be getting back to you in the next week.